It's a pleasure to be here. It's been actually many years since I've been up the coast of Haifa, and I can thank Zev and Shraga for the opportunity to visit and also to present the lecture this morning. So as Zev said, my talk will be in the area of cancer chemotherapy and drug metabolism, and I will speak about two aspects of our work. The work that we've been doing the last 10 years or so in the area of gene-directed prodrug therapy, whereby we try to improve the selectivity for anti-cancer drug action by facilitating the activation of a prodrug directly within the tumor cell target using gene therapeutics. And I'll tell you a little bit about that. Then I will transition, and you will see the natural transition that our research took to the second area that I will speak about. And this is targeting tumor-associated endothelial cells using anti-angiogenesis and the intrinsic conflict that we have combining or making efforts to combine anti-angiogenic drugs with traditional chemotherapy. So I came to this field many years ago when I was at the Dana-Farber Cancer Institute because of our interest in the cytochrome P450 enzyme system and the major role that it plays in drug metabolism, which generally leads to drug deactivation and drug elimination. However, in a select number of cases, pharmaceutical drugs are actually prodrugs. That means they're inactive precursors that require metabolism for their enzymatic activity. And as such, we can take advantage of the metabolic properties of the enzyme systems in an effort to improve the chemotherapy. So the overall goal of cancer pharmacology can be viewed as to improve the poor therapeutic index that most anti-cancer drugs, classic anti-cancer drugs, have. And you'll excuse the font size change that comes with the 7-hour transition across the Atlantic. So if you can read, I don't write like this, but sometimes it comes out. One goal would be to tailor the choice of drugs and dosages based on each individual patient's profile for cytochrome P450 enzymes, which vary markedly due to pharmacogenomic and genetic factors, and also vary markedly because of recent history of exposure to other medications, for example, in combination chemotherapy, many of which will themselves alter the pattern of gene expression in liver and in other tissues. Another approach, which is the one that we've taken, is to try to modulate drug metabolism and drug targeting in an effort to increase therapeutic activity but not increase host toxicity. So the paradigm is shown here in diagram form. If we have a population of tumor cells which are generally deficient in P450 enzyme activity, and what we would like to do is, through gene therapy, introduce the therapeutic gene, which in this case would be an enzyme that will activate a prodrug. But the limits of the technology are such that we can only effectively transduce a few cells, shown here, in an overall tumor cell population. So in order for this to work, what we require is something called bystander cytotoxicity, whereby the activated metabolite has the chemical and pharmacological capabilities that it can diffuse out and kill other tumor cells nearby. And so we've exemplified this, and we've developed this, and as I'll show you briefly, it's now in clinical trials, taking the anti-cancer drug cyclophosphamide, Cytoxan, which is a well-known, long-standing chemotherapeutic agent. It undergoes P450 metabolism to activate the drug, ultimately to phosphoramide mustard, which is a DNA cross-linking agent. And the primary metabolite, 4-hydroxycyclophosphamide, when produced, is normally produced almost exclusively in the liver, but can then gain access to other tissues through the bloodstream. It has intrinsic bystander activity that can not only go to adjacent cells, but to more distant cells. So the question that we asked now a good number of years ago, whether introduction of a P450 enzyme that will activate cyclophosphamide, introduction into a tumor cell, will sensitize the tumor cell to the drug. And this is shown in a model system here. 9L is a rat glioblastoma cell line that we've transduced with a marker enzyme, LAC-Z, so we can stain those cells blue within the population, so we have a mixed population of cells. Or alternatively, we can transduce the cells with a P450 gene, the human P450 gene, encoding cytochrome P450 2b6, which can activate cyclophosphamide. And what you can see is that in response to increasing concentrations of cyclophosphamide, there's no effect on the culture in the absence of P450, exemplifying a typical human tumor, whereas in tumor cells that have been transduced with retrovirus encoding the human P450 enzyme 2b6, we can kill the cells in a dose-dependent manner. But what you see is we kill not only the blue cells, but we kill the bystander non-blue cells in the population. And this is a reflection of the bystander cytotoxic response associated with the intrinsically diffusible cytotoxic metabolite. We considered various P450 enzymes from the same family but from different species, and what we found is that the human enzyme is not the best enzyme in terms of its enzymatic and metabolic properties. And in a collaboration with Jim Halpert's lab, then in Galveston, Texas, who had developed panels of site-specific mutants of these various enzymes, we determined that a particular constellation of amino acid residues at the active site of the heme protein cytochrome P450 was more active with these particular mutations, at which point we recognized that there's a naturally occurring P450 enzyme that has these same particular ideal amino acids, and that is the canine, the dog enzyme, cytochrome P450 2b11, which is characterized by a low Km. And if you think back to your basic biochemistry, that means that this enzyme can efficiently metabolize, can activate the anti-cancer drug at low concentrations. And you can see that over here. The human enzyme has a Km of more than 1 millimolar, and you can put that in context. Maximum blood levels of the drug in a typical cancer patient after treatment would be on the order of 1/10 of that, 100 micromolar or even less. Whereas the dog enzyme has a Km about 20-fold lower, more favorable. And as a consequence, in the cell assay that I showed you, the parental tumor cells are insensitive. The cells that have been transduced with retrovirus bringing the human enzyme are sensitive to about 200 micromolar, whereas the dog enzyme gives about 100-fold improvement of the anti-cancer activity. And an in vivo model, in a subcutaneous xenograft model, we can see that cyclophosphamide treatment leads to a very modest growth delay. This is at a maximum tolerated dose in the animal model, whereas introduction with the retrovirus of the human enzyme delays by 21 days growth and a more substantive delay of 32 days with the dog enzyme. So really the dog enzyme would be the best way to go forward. Clinical trials that are currently underway were started, of course, before our observation that the dog enzyme is the better enzyme. They use the human enzyme. This is a breast cancer trial with 10 late-stage breast cancer patients and also mixed in 2 melanoma patients with accessible skin nodules, intratumoral injection of the therapeutic gene, which is called METZIA, which is a retrovirus that has the 2b6 linked up to a LAC-Z marker to phenotype the patients in terms of the efficiency of gene delivery in vivo at doses that were shown to be safe, followed a week later by oral cyclophosphamide, and then a second cycle, and then evaluation of safety and gene transfer by biopsy and assessments of any indications of efficacy. And these are studies that were sponsored in the UK by Oxford Biomedica, a gene therapy company. And the most exciting result from this and a subsequent phase 1 trial are summarized here, that not only was the responsiveness in individual patients of the treated lesion, but there were apparent responses of distal lesions, suggesting that there was a systemic improvement in response to localized delivery of the therapeutic P450 gene. And that was associated most dramatically in these 2 patients, but also in 3 other patients with the activation of an apparent anti-tumor immune response in those patients, these particular antigens being highly represented in these 2 patients, and the pretreatment levels of antigen reactivity were nil and were induced in a time-dependent manner. And this therapy is currently being investigated in a phase 2 trial. So what we can see is that cyclophosphamide in this paradigm of prodrug activation gene therapy can kill tumor cells by 2 types of mechanisms. One, by a tumor cell bystander killing, whereby the drug is activated within the tumor cell and then can kill the tumor cell or adjacent tumor cell. But also we have the potential for a systemic immune-based killing mechanism that's potentially even more exciting. And we can speculate that it's associated with release of tumor antigens in a strange and unusual way when the tumor is killed from within, as opposed to with drug coming from outside the tumor, derived from the liver. And you'll see the connection with some of the work that I'll show you in a little while. There are other potential mechanisms in terms of release of host factors or tumor factors in response to cell killing. So we began a number of years ago to investigate what was at the time a brand new concept of metronomic chemotherapy that was introduced by the late Judah Folkman almost a decade ago now. And the observation that was made is that endothelial cells lining the tumor vasculature are intrinsically sensitive to low concentrations of cyclophosphamide, of activated cyclophosphamide. In fact, they're more sensitive than tumor cells themselves. And so the observation was that if you treat with a typical classical maximum tolerated dose chemotherapy, give as much as you can to kill as many tumor cells as possible, then wait for the patient's sensitive critical host tissues like bone marrow to recover, and then come again and another cycle and another cycle. If you replace this with a cycle that was empirically determined in animal models to be best at every 6 or 7 days, a lower dose, the idea is that you will prevent the recovery of the endothelial cell compartment. And as such, you'll induce the expression of anti-angiogenic factors, and now the chemotherapy will have 2 effects, according to the idea. It will have an anti-cancer effect combined with an anti-angiogenic effect. So the first thing we tried is we said, can we put this together with the gene therapy model? So this shows the growth rate of individual tumors, the same 9L gliosarcoma in vivo. And the animals are killed here because the tumors are getting too large. And this shows when we combine with the metronomic chemotherapy. And what you can see is there's major regression, which we had never been able to achieve before with the maximum tolerated dose schedule. There's major regression of this tumor cell line. At this point, we halted chemotherapy. We halted the metronomic cyclophosphamide, and what you see is that there's immediate rebound. So the tumors are reduced to a small but measurable size, but the cells are perfectly viable. As soon as you withdraw the metronomic chemotherapy, they take off. These are highly vascularized tumors, and it's virtually impossible to kill them altogether. Now what happens if we introduce the P450 gene with the retrovirus, either the human 2B6 or the more efficient canine 2B11? What you can see is a few things. First of all, the tumors don't get quite as large before they regress. Second of all, we now have a period of time, about 30 days, where we can't measure the tumors at all. But nevertheless, ultimately, the tumors will regrow. In the case of 2B6, in the case of 2B11, we have an extended period of time, 79 days, where there's no tumor measurable. And now we have a few tumors, but not all the tumors that begin to regrow. And so the idea is that we're killing the tumors much more efficiently with this paradigm, because in addition to having activated drug generated in and coming from the liver, we have activated drug generated within the tumor cells, within the tumor themselves. Another important feature to pay attention to is we begin treatment on day 0, and the relative tumor volume is set to 100% at this point. Look how large these tumors get before they begin to regress. they grow 5-fold in size. In this case, they're growing about 3 to 4-fold, and similar over here. So this therapy, this metronomic therapy, has its advantage, but a disadvantage is that the tumors continue to grow for some time before they respond. It takes several cycles of the metronomic chemotherapy for the response to kick in, which is consistent with an anti-angiogenic mechanism. First, you have to kill off the vasculature, and then you have to have the secondary consequence of anti-tumor effect. But as I'll show you a little later on, we believe that that is not the major mechanism for this regression. Okay, so this is our vasculature feeding the tumor with a maximum tolerated dose schedule. The liver-derived 4-hydroxycyclophosphamide can get in and can kill off the tumor cells, whereas with a metronomic therapy, we have this, plus we have the intratumoral generation of the activated drug, and this is the traditional anti-angiogenic component, so we've now regressed not only the tumor cell population, but also the vasculature. And the question is, does intratumoral P450 enhance the anti-angiogenic response? So you would think, misechel, of course it does, because it kills the tumor cells. But sechel is not always the right answer. So we investigated the question, and it's done very simply. We look at the effectiveness of the anti-angiogenic response in tumors that don't and tumors that do express the P450. And this is immunostaining for CD31, which is a marker for endothelial cells. And what you can see is that as a function of time, there's a loss of tumor vasculature, and it's quantified here by FACS analysis. But the loss in response to each cycle of metronomic chemotherapy is equivalent, independent of whether the P450 isn't or is present. That is to say, the anti-angiogenic response is unaffected by the presence of the P450 enzyme. And so the view is that metronomic cyclophosphamide has significant anti-angiogenic component because we see the marked loss of the endothelial cells associated with the tumor. However, intratumoral P450 contributes in only a limited way to the ultimate anti-angiogenic response. To be effective, then, the metronomic chemotherapy requires a mechanism that ultimately targets the vasculature, and that we believed is exclusively or primarily derived from the action of the drug that is coming from the outside of the tumor. But we also require the Trojan horse effect, the activated drug generated within the tumor. And so here's the view. We have tumor bystander killing that I've shown you before. But we also have liver-derived 4-hydroxy, which is coming from the outside and killing the tumor. And the reason we think there's a difference between these two sites of generation of active drug relates to the nature of the tumor vasculature. But the blood flow derived from the outside allows the activated drug to get access, efficient access to the tumor vasculature, but does not allow the drug to get efficient access to the tumor cells themselves because of the tortuous nature of tumor vasculature. And just the opposite, the drug that's activated within the tumor has good access to the tumor cells, but poor access to the tumor vasculature. Okay. So the next question we asked, and this is a transition, is whether the anti-tumor response can be further improved by combination with bona fide anti-angiogenic drugs, not cyclophosphamide combined with itself, but rather combined with VEGF receptor-directed anti-angiogenic drugs that have been the hallmark of new drug development in cancer over the last several years. So VEGF is important for tumor angiogenesis. Tumors feed upon themselves by VEGF-stimulated growth of vasculature. And as such, this has become an important therapeutic target for cancer. These are 3 recently approved, last few years, clinically approved agents: Avastin, which is an antibody against VEGF, Sutent and Nexavar, which are small molecule receptor tyrosine kinases that will target the VEGF receptor. So this shows you what is the best that one can do clinically with these agents, more or less today. It's good, but really not. There is an improved response that is seen, but the median survival benefit is only several months and does not persist. Moreover, in most cases, there's a requirement for combination with chemotherapy to observe efficacy. So the responses are rather poor in the final outcome for the patients. So we've been looking at an investigational VEGF receptor inhibitor, a Pfizer compound called axitinib, whose structure is shown here. This shows that in our 9L tumor model, this drug can efficiently decrease microvascular density, and similar to patients, it gives some growth delay but no therapy, no ultimate therapeutic activity. So we tried combining this with metronomic cyclophosphamide, and we have a surprise. So this again is the data showing a small growth delay with the anti-angiogenic drug itself. The green curve shows you the response to metronomic cyclophosphamide alone, whereas the tumors continue to grow after we initiate therapy, just as I showed you earlier, and ultimately they regress. And in this case, there's the P450 presence, and so the tumors can be eliminated. But what happens when we combine these two therapies? So there are two observations. First is that the combination is immediately tumor static. The tumors literally stop growing by the first measurement 3 days after we initiate therapy. And that's an improvement over the metronomic cyclophosphamide alone, where in this case the tumors approximately double before they begin to respond. So that's good news for the patient. I presume you don't want to have a patient whose tumor gets twice as large after you start therapy and you say, don't worry, eventually it will regress. But the other aspect of the therapy is that the combination eliminates the ability of metronomic cyclophosphamide to regress the tumors. And that's very disappointing. And so that's what I want to spend the rest of our time discussing. Why is that and what's happening? So is the drug working properly, and what are its effects? So the anti-angiogenic drug axitinib reduces tumor perfusion. That's what it's supposed to do by closing down blood vessels and causing their loss, causing their destruction. So we can measure tumor perfusion, for example, by giving a test dose of cyclophosphamide and then monitoring the 4-hydroxy metabolite in various tissue compartments. And you see the plasma compartment is unaffected by drug treatment, by the anti-angiogenic drug. Levels of the active metabolite in the liver unaffected. That's as expected. And we can see that within 12 hours of a single dose of the anti-angiogenic drug, that we've partially closed down the tumor vasculature, as expected. And the result is that there's a decrease in penetration of the active drug formed in the liver getting into the tumors. So these are tumors that are not undergoing the P450 gene therapies. So the drug, to simplify it, is coming exclusively from the liver. Furthermore, there's the expected increase in tumor hypoxia, which is stained here with this dye, in response to the anti-angiogenic therapy. And there's a decrease in the proliferation of the tumor cells. So all as expected. But we can put that together to say that once we treat with the anti-angiogenic drug, the blood vessels feeding the tumor are now thinner, they're closed down, and ultimately there are fewer of them. So there's vasoconstriction due to the block of the VEGF pathway, which generates nitric oxide. And that results in decreased penetration of the active drug into the tumor. And there's also a decrease in the proliferation of the tumor cells, and that renders them intrinsically less sensitive to the drug. But is that enough to explain the inability of the drug to regress the tumors? Because the decrease that you saw was only on the order of 30 or 40%. So another observation that we made is that a protein factor, thrombospondin 1, which was characterized as an anti-angiogenic factor that's important for the regressive effects of metronomic cyclophosphamide, this factor is induced by metronomic chemotherapy within the tumor cells, and that induction we observed is blocked by the anti-angiogenic drug. So we said, could that be important? And maybe the axitinib metronomic cyclophosphamide combination is no longer anti-angiogenic. Maybe that's the problem. But that's not true. Anti-angiogenesis is still decreased, blood vessel density, and the pericyte coverage of the blood vessels is still reduced. and also the hypoxic marker, in this case GLUT1, is induced independent of whether the chemotherapy is present. So by these measures, the combination looks the same as axitinib treatment alone. So there's decreased blood vessel density and increased hypoxia. They're still observed with the combination. So what is the problem? So a paper appeared late in 2008 showing that overexpression of this factor, thrombosbondin 1, induces another factor, PAI1, that's involved in recruitment of macrophages to tumors. So we got the idea from this paper that perhaps macrophages are involved, are important, because we know that the metronomic cyclophosphamide can induce this factor, and perhaps there's some macrophage involvement. So what you can see here are immunostaining of the tumor with a macrophage marker, and you can see that metronomic cyclophosphamide is able to induce the recruitment or activation of macrophages within the tumor, and the combination with the anti-angiogenic drug blocks that response. Now, what type of macrophages are these? There are, to a first approximation, we'll consider two types of macrophages that have been designated M1 and M2. M1 are anti-tumor macrophages that do all sorts of good things, including tumor cell lysis and activation of certain chemokines and metalloproteinases. But there are other macrophages that are bad macrophages. There are pro-tumor macrophages, the M2. These are pro-angiogenic, and they induce tumor growth. And there are various markers, and so we considered two markers in combination, arginase 1 and NOS2. And what you can see here is that the anti-angiogenic treatment is strongly inducing the ratio of the bad to the good macrophages, the M2 to M1 ratio. Metronomic cyclophosphamide is not, and the combination of drug treatment still induces them. And that's achieved by inducing the pro-tumor macrophage, the M2s, and by suppressing the anti-tumor macrophages, M1. So the anti-angiogenic drug, at least in this model, is looking to be not so good in this regard. Well, at this point, we considered other types of innate immune cells, which include natural killer cells and dendritic cells, in addition to macrophages, as to see whether they're affected. And what we observed most strikingly with the natural killer cells is that metronomic cyclophosphamide will activate these cells, will induce them within the tumor, and this response is blocked by the anti-angiogenic drug. And this is in one tumor model, one brain gliosarcoma, the human tumor cell line U251, and this is the rat9L gliosarcoma. And you can see over here is a time course of metronomic cyclophosphamide treatment that's induced, and that induction is blocked by combination with the anti-angiogenic. And natural killer cells are known to kill tumor cells by binding to them with various receptors and by secreting factors that will then kill the adjacent tumor cell. And two of the factors that are very important for this killing mechanism involve the macrophage-derived protein perforin-1, which perforates, forms a complex in the cell membrane of the tumor cell to allow for granzymes formed by the macrophages to enter the tumor cell and to effect tumor cell killing by activation of caspase activities within the target tumor cells. So we looked at the granzymes, and what you can see here very clearly is that there's a dramatic induction of granzyme A and granzyme B by metronomic cyclophosphamide, and that response is totally blocked by the anti-angiogenic. The perforin-1, which forms the pores on the tumor cells, is also induced by the metronomic chemotherapy, and this is blocked by the anti-angiogenic, as is FAS, which is an important receptor protein in mediating the interaction between the macrophage, between the natural killer cell and the tumor cell. So what are ideas as to how is this whole process happening? So we don't really know. We're just beginning to investigate this. But if you look at various chemokine and cytokine gradients that are put out by tumor cells and that can attract various innate immune cells. These signal direct mobilization of the immune cells. And if we look at chemokines that have been associated with the recruitment of macrophages or NK cells, specifically interleukin-12 beta in the case of the macrophages, or the chemokine CXCL14 in the case of NK cells, what you can see is the same pattern that I've been showing you before. Activation by metronomic chemotherapy and a block in that activation by the anti-angiogenic drug. So this takes us to the close and the model. And what we observe is that metronomic cyclophosphamide, this is the schedule that we use in the animal model, every 6-day treatment, will lead to activation of cyclophosphamide in the liver, or in the case of the gene therapy model, in the liver plus within the tumor. The drug will gain entry to the tumor, will kill tumor cells, and there will be signals that are put out by the tumor in association, in response to this activation of a killing mechanism that recruit various innate immune system. All these experiments, I should mention, are carried out in a SCID mouse model. So T and B cell responses are deficient, but the innate immune cell system is wholly intact. These factors then infiltrate into the tumor and or are activated, and we believe that this is the major mechanism, we hypothesize that this is the major mechanism whereby these very large tumors are regressed. We can also speculate that in the P450 gene therapy trial that I showed you, where there is activation of an immune response, in that case it wasn't an innate immune response that was probed for, but then again those are immune competent patients as opposed to our immune incompetent mouse patients. We can speculate that this is in fact a contributor to the mechanism for the anti-tumor response observed in those patients. Now, what happens with the anti-angiogenic drug? Why does that, and how does that interfere with this process? So at this point we don't know, but we have many hypotheses based on our now retrospective careful reading of literature, which shows the importance of VEGF receptor signaling for a number of processes. So we already know from our own work and work of others that anti-angiogenic drugs will slim down the tumor vasculature and make it more difficult for the drug to get in. So that's one point of action, but we believe that can't be the whole story. In terms of signals that the tumor may emit and cytokine gradients, from our work it's clear that if these various chemokines are indeed important and critical, that they are no longer produced in the presence of the anti-angiogenic drug. And there are various reports of activation of innate immune systems requiring VEGF receptor signaling. So these are some of the ideas as to how the VEGF receptor may be important for not only endothelial cell, but for immune homeostasis and for progenitor potentiation and mobilization. So in conclusion, metronomic chemotherapy will induce host innate immune system recruitment, and this we propose may facilitate tumor regression. And we have on plans now for direct testing of this using a more complete immune deficient mouse model that's deficient, in particular in the NK and macrophage system, that is to say the SCID-NOG mouse model. This, we believe, involves induction of chemokine and cytokine gradients for guided mobilization of the innate immune cells to cyclophosphamide-treated tumors, with recruitment of macrophages, natural killer cells, and I didn't show you dendritic cells. And this action of the natural killer cells involves increase in activation of the granzyme perforin pathway and other cytotoxic immune effectors. The anti-angiogenic VEGF receptor selective tyrosine kinase inhibitor axantinib suppresses these innate immune responses. Most notably, it blocks NK cell recruitment and it shifts the tumor macrophage population to a pro-tumor macrophage subtype. Now, one word of qualification, which the people at Pfizer said, please, make sure you make this point. Everything that I've showed you is based on the types of studies that I've presented. We don't understand the underlying mechanism. We need to do more to elucidate that. We've looked at only 2 tumor models with respect to this response. They're both highly vascularized gliosarcomas. We've looked at them in a subcutaneous model. We haven't looked at them in an intracranial model. We haven't looked at various orthotopic tumor models. We haven't looked at other anti-angiogenic drugs. This may be a class effect of the anti-angiogenesis as opposed to particular to this anti-angiogenic drug. And so these are qualifications. And certainly the clinical relevance of these studies is really unknown. But nevertheless, with those qualifications in mind, this and other observations unrelated to this, for example, showing that anti-angiogenic drugs can induce tumor metastasis, which is a very, very big concern, raises some questions as to this class of new drugs. How do they work? Are they beneficial? Or my view is, what do we need to do to harness their beneficial effects and minimize their potential for deleterious action? And along those lines, we are pursuing, and others are pursuing, studies to try to identify what are the logical combinations and therapies with chemotherapies to try to minimize the bad side and to improve their actions. So let me acknowledge some really outstanding people in my lab who've contributed to this work. This is our cancer therapeutic and anti-angiogenic group. Jay Ma is a recent Ph.D. graduate from my lab who's currently doing a postdoctoral at Children's Hospital in Boston. He carried out all of the early studies on the anti-angiogenesis that I showed you. And Josh Doloff is a current Ph.D. student who is working on all of the work with the axatinib combination and the innate immune system recruitment. So that is entirely Josh's work. And these are other students and postdoctoral fellows who are working on other aspects of our project that I didn't show you. And the earlier work on the P450 gene therapy is primarily carried out by a slew of earlier fellows and students in the lab. I'll thank also Danny Hulot of Pfizer for collaborative work with axatinib, Jim Halpert, now at UCSD, with whom we carried out the initial studies discovering the 2B11, and Stuart Naylor and others at Oxford Biomedica who've carried out this cytochrome P450 gene therapy trials, and of course the NIH for support. So I'll stop here and be happy to answer questions.